So we have discussed epidemiology, which is the spread of an infection in a population. And again, it was population dynamics. It was susceptibles and infecteds, and susceptibles bump into infecteds, and it's the same shark-tuna dynamics. Basically, population dynamics. The population dynamics of animal populations or the population dynamics of susceptible and infected people. Then, new subject. 25 years ago, a zoologist and mathematical modeler named Angela McLean, who was at Oxford, realized something. Let's discuss a completely new subject. I want to discuss immunology. That is the branch of medicine that studies the immune response within the body. And I'm giving it away, the population dynamics of various immune cell species, like T cells and B cells and helper B cells and killer T cells. And these are populations. And they are manufactured in the bone marrow, and they have various things in place to keep the populations up and to control them. And you have these populations of cells in your body. And then, let's say, everything is going along very nicely. And then something introduces an HIV virus particle into this system. So now we are looking within an individual. So you see the title, Population Dynamics of HIV Within an Individual After Treatment with Zidovudine or AZT. So the idea is you have populations of cells. You have T cells, and now you have another population, which is V for viral particles, virions, as they're called. So what's the fundamental event here? It's V meets T, or T meets V. T cell meets virus. And the result of that is that the virus infects the T cell with probability beta. And again, the same concepts apply to this beta as to the beta in the population, human population dynamics case, where it had to do with safe sex practices. But here, well, it also had to do with the possibility of drugs inhibiting that process. And that's closer to what we're talking about here, because V meets T is going to result in the infection of that T cell with a probability beta. And drugs can alter that probability beta. So here is her model. And you see 4 state variables. R is the lymphocytes that are actually infected by the HIV virus. They're called CD4 positive lymphocytes. And those R lymphocytes bump into V's, viruses. When they do, a fraction rho of them become latently affected, which means they're just kind of quietly infected and not expressing virus themselves. But the rest of the fraction, 1 minus rho, become expressively infected And they start producing, those are the T cells that start producing the extra viral particles, which is what is going to give you the disease. So how often does an R bump into a V in your body? The answer is the product RV. And that's multiplied by the coefficient beta, because beta is the likelihood of an R meeting a V, resulting in the infection of the R and the production of more V. So their model is very straightforward. You could basically write the equations from staring at the diagram. And they generated a number of very interesting predictions about the rise and fall of the viremia post-HIV infection. And then just as another example, we were able to use that model to answer a very simple and very practical question, which is our docs at UCLA in the neonatal intensive care ward have to deal with neonatal HIV, that is maternal fetal transmission of HIV. And now you're in an interesting situation illustrated by this cartoon, which is the infant is born on day 0 with no immune system. You begin to acquire antibodies as you begin to get exposed to the environment. So the infant is born, and directly the infant starts developing its immune system. But as the infant is developing its immune system, it is under attack from the HIV virus, which is in turn under attack from you. Now, all of these variables are going to be feeding back on and affecting each other. And so that creates a certain problem of how do we proceed? What kind of dose do we give? What kind of time course of dose do we give? Because it's not just going to be a single shot. What kind of time course can we give? And the important thing is that all of this is going to be changing out from under us. And we have another major constraint. We can't simply give lots of the drug because we're dealing with a neonatal infant. And in all drug therapy, infant, adult, whatever, we have this problem. It's called the problem of the therapeutic window. So you have varying doses of drug that you can give, from low on the bottom to high on the top. And very low doses of the drug are simply going to be ineffective. Very high doses of the drug are going to be toxic. In between those two is the magic zone that is called the therapeutic window. And we have to find it. We have to find the parameters that define the therapeutic window and find their value. And it's further complicated by the fact that in this case, because of the developing infant and your therapy, the therapeutic window is changing in time. So you have to hit that therapeutic window, which is changing in time. This becomes a little bit complicated. So our HIV neonatal docs said, is there something we can do? And Paul Krogstad and Yvonne Bryson, two wonderful HIV docs, understood the importance of modeling. And so they said, this is too complicated for us to sit with our feet up and say, well, I think we should give a lot of this and then stop and give more of that. That's not going to work. So armchair theorizing about dosing is not a good idea here. And they grasped that totally and said, let's make a model. So we did. Here's the paper that we wrote. But what we used was basically Angela McLean's model of viruses and T cells and activated cells and inactivated cells. We did add one term to the McLean model of inactive T cells and activated T cells. We added that into the model. We now have a 5-variable model. And you see the 5 differential equations. And they're kind of exactly what you expect from the compartmental model. Quiescent cells are produced at a rate T. They die at a rate tau. All of the terms, and you should be able to stare at that equation and go through it term by term and tell what every single term is doing in that model and why, given what it is supposed to be representing, why does it have the shape that it does? Just for example, you see that in the E prime equation, the expressively infected cells, you see a plus alpha L. You also, in the L prime equation, you see a minus alpha L. So the same term appearing negatively and positively is the sign of stuff being subtracted from this compartment and being deposited in that compartment. And that rate alpha is simply the rate at which L cells go to E cells. Latently infected become expressively infected. That is the rate alpha. So you should be able to go through every single one of the terms in this model and say, why does it have the form that it does? And what kind of effect does that term represent? So then once we had the model, we were able to do some simple experiments. For example, let's look at the left-hand side here. This is an experiment where we tried 3 different sizes of the initial inoculum. So let's just ask a question. If you got 10 HIV cells injected into you, or 100 HIV cells, or 10,000 HIV cells, would that make a significant difference in the course of the disease? And the model, as you see, says no, not really. The 3 different initial conditions, differing by many orders of magnitude, the 3 different initial conditions all go to the high value, and they all go there fairly quickly and stay there, but the bigger the inoculum, the faster, and by faster I mean by a week or 2, is the progression to full-blown viremia. So initial inoculum doesn't make a big difference. Then we did some beta experiments. Remember, we're looking for the parameters that define the therapeutic window. So here are 4 experiments with beta, and you say, I only see 3 of them. For beta equals 005, beta equals 004, you still get the rise to the full-on viremia, and it stays high, but it takes a little bit longer. And beta is 003, same thing. When we set beta equals 0001, however, the result goes to 0 and stays there. In other words, there is your therapeutic window. The therapeutic window is between beta equals 0001 and beta equals 0003. You can tell from lab experiments what your beta is by seeing how effective it is at preventing the infection. And this gives you the actual numerical parameters of the therapeutic window in neonatal HIV.